Hello everyone, this is Dr. Lalit Mittal from Giggles Clinics and my own space. So today's video is another important video about the role of gut microbiota and its importance and its effect on future health. So in this video, I'll be describing in brief about what are the factors which are responsible for the microbial diversity, improving the gut microbiota and its effect on future health. So a brief, what is gut microbiota? We have been talking a lot about gut microbiota, gut dysbiosis. So what is this gut microbiota? Microbiota is a term which refers to a collection of different types of microorganisms which are present in our body. It may be bacteria, it may be viruses, it may be protozoas, it may be fungi. So this whole collection of the genetic material of these different types of organisms which are present in our body refers to as microbiota. Now it is very interesting to note that the ratio of the DNA microbiota is 10 times more than that of the human DNA. Means the ratio is 10:1 compared to the bacteria DNA to that of the human DNA. So we can imagine that we are more of a bacteria than a human body. Now, what are the factors which are responsible for governing this gut microbiota, or what we call as microbial diversity? That is the different types of microorganisms which are present in the gut and which maintain a harmony between the good and the bad bacteria. We know that there is always beneficial bacteria and there are always non-beneficial or pathogenic bacteria. They always remain in harmony. Now, when the balance between the two gets disturbed, what will result in what is known as gut dysbiosis, that is the predominantly the bad bacteria take on the beneficial bacteria. So there are three most important factors which govern the microbial diversity in the human body. The first one is intrauterine factors. Now intrauterine factors, or what we call now by the term intrauterine hypothesis. Intrauterine means inside the womb. When the mother conceives and the child is there in the womb, that period before the delivery is known as intrauterine period. Now till now or till recently it was thought that intrauterine period or intrauterine environment is sterile, means it doesn't contain any bacteria, any pathogenic or any harmful bacteria. As a result, the diet, the type of food which the mother is taking would not affect the intrauterine environment, as it is sterile, free from microorganisms. But this theory over a late period it has been challenged, and over animal studies, over the mice, it has been found the intrauterine environment is not at all sterile. In fact, it contains both the pathogenic and the beneficial bacteria. So this means to say that whenever the environment in the intrauterine environment is disturbed, the pathogenic bacteria may take over than the beneficial bacteria and may result in disturbing the gut microbiota of the mother. So the type of food the mother is taking, the amount of nutrition, the amount of carbohydrate, fats and proteins, iron, folic acid supplementation, the environmental stress to the mother during the pregnancy. It may be environmental stress, it may be family stress, it may be mental tension, or it may be the infections like UTI, high blood pressure. All these factors, they affect the genetic material of the microbes, or what we call as epigenetics, that is the environmental stress which is having on the genome. So that factors may cause the disturbance or in the phenotypic or the expression of the genetic material of the microbes in the human body in case this mother, and as a result may disturb the gut microbiota or microbial diversity. So this means to add that the intrauterine environment plays a very important role in affecting and governing the microbial diversity, which earlier it was thought to be sterile. Now the second most important factor is the type of delivery. Now generally the children either may be born by a normal vaginal delivery or by the cesarean section. But in present world, in present scenario, many kids are being delivered through cesarean section. Now those children who are born through cesarean section, the probability of going to microbial diversity or microbial disbalance will be probably will be higher compared to the children who are born through the normal delivery. The reason is because when the child is born through a normal delivery, it has been exposed to the vaginal canal, and that is consisting of large amount of beneficial bacteria like lactobacilli and bifidobacterium. Whereas those who have been born through a cesarean, they have been exposed to the microbiota, or they have been exposed to the bacteria which are present on the skin of the mother, and these are considered to be pathogenic, like Staphylococcus, Streptococcus, etc. As a result, hence it is believed that those children who have been born through normal delivery, the chances of going to microbial dysbiosis will be less compared to those who have been born through cesarean section. And the third most important factor is the type of feeding, whether the child has been exclusively breastfed in the first 6 months, whether it has been formula fed, and what was the time of complementary feeding, that is the time when we start on giving the solid feeds. Now, there is a very peculiar property of the human breast milk. Now, the breast milk contains some specific materials which are known as HMOs, which are known as human milk oligosaccharides. Now, they are prebiotics, and they act as prebiotics for the growth of beneficial bacteria like bifidobacteria and lactobacilli. Although nowadays the formula feed, they do contain these lactobacillus reuteri, lactobacillus ramesis, and also the plant-based prebiotics like FOS and GOS. They are also act as prebiotics, and they also help in influencing the growth of bacteria. But compared to HMOs, they don't influence the growth of specific beneficial bacteria like lactobacillus and bifidobacteria. Another important thing is the HMOs, they are three-dimensional structure, hexagonal, whereas the FOS and GOS, they are linear structures. Now, because of these two peculiar properties between the HMOs and GOS or FOS, HMOs are considered to be at a much higher beneficial compared to GOS in influencing and promoting the growth of beneficial bacteria like lactobacillus and bifidobacteria. So means to say, just a bottom line to say is that those children who have been breastfed have a better chances and better advantage of not going into allergy conditions and helping in maintaining the microbial diversity, because this microbial diversity, this gut dysbiosis, if it is disturbed, the microbial diversity is disturbed, resulting in gut dysbiosis, can lead to a lot of allergy conditions like asthma, diabetes mellitus type 1 in children, recurrent sore throat infections, asthmatic problems, dermatitis, skin problems, eczemas, and in future may also lead to obesity. So as a result, if we are able to maintain the microbial diversity in the first 1,000 years of life, we can definitely influence the future health of our children in adult, because the 2 to 3 years of life is very crucial. During this period, there is a lot of microbial diversity takes place, because during the first 1 to 1.5 year, the microbial diversity is different compared to a child when he reaches to the age of 2 to 3 years. By the age of 2 to 3 years, the microbial diversity is equivalent, is similar to that of the microbial diversity in the adult. Means that by the age of 2 to 3 years, the amount of bacteria or the type of bacteria which will be present in a child will govern the type of bacteria which will be present in an adult. So hence, if the beneficial bacteria are predominantly present like lactobacillus and bifidobacteria in the first 2 to 3 years of life, then definitely the gut of the child when he goes into an adult stage will be definitely better, and hence he will be less exposed to diseases like coronary artery disease, heart diseases, diabetes, blood pressure, asthmatic problems, allergic issues. So this microbial diversity is influenced by nutrition, the mother nutrition, the type of nutrition the newborn is being taking, and also the third most important factor is complementary feeding. Now, why this complementary feeding is being given at the age of 6 months? Why it cannot be started at 4 months? Why it cannot be started at 8 months? Complementary feeding is the period when we start introducing solid feed to the newborn by the age he reaches to 6 months. Now, there is a logical reason for that. Now, the first 6 months, the child has been exclusively breastfed, because the protein requirement of the child during that first 6 months is around somewhere between 1.5 to 1.7. Now, this protein requirement is absolutely fulfilled by the breast milk. It is apt for this range. But when the child crosses the age of 6 months, the protein requirement increases, and which cannot be taken care by the breast milk alone. Hence, we advise the mothers to start breastfeeding, to start complementary feeding the child, introducing solid feed, so that his excess energy requirement in the form of protein, fats, and carbohydrates can be fulfilled by the protein, can be fulfilled by this complementary feeding. So this is the importance of introducing complementary feeding at the age of 6 months and following exclusive breastfeeding. So in this video, I have described about some of the most important factors which can govern the microbial diversity, and as a result, we can help in preventing the future allergic problems, illnesses, obesities in our kids once they reach an adult stage.